An example I often use is for the artificial heart, they actually picked a ladies' girdle material because it had a good flex life. But that would lead to problems. And like when blood hits the surface of that artificial heart, it could form a clot. The clot goes to the patient's brain, they get a stroke and they die. And I started thinking, rather than take materials from your house, you could really use chemistry to design materials from scratch that would have better and better properties. That ultimately would lead to new brain cancer treatments.
We're just going to do the last session of the day today, the hero of science, Robert Langer. As I shared with you, there are 3 or 4 speakers who've not been able to join us the way they intended. Robert Langer was willing to be like a Concorde, you know, leave on a Friday, arrive on a Saturday, and get back on a Sunday. If we could bend time enough to get him there, we tried, but we couldn't because something very urgent came up for him. But he's still joining us on Zoom. We didn't want to leave that because his story is truly one of those unadulterated celebrations of science, a wonderful note to end this first day on, much like David Puccino, just stretching the boundaries of humane technology and science, improving our lives, no debates on that. The hero of science, Robert Langer. Ladies and gentlemen, I'm going to be joined on this conversation with Ajit Singh, who's Robert's friend and also a white space tech investor in the health space. And I'd ask you to join me in welcoming Ajit and Robert Langer onto the stage.
Bob, we met about 9 months ago. I was at the Convergence Forum in Chatham, Massachusetts. And
Right.
You were being interviewed by Anna Kuchman from Boston Globe. And taking any narrative from mass media will be too reductionist for a person with talents and accomplishments as broad as yours, but I'll start there. So that was an interview where they were extolling the virtues of Moderna and your work. And of course, in mass media, you're predominantly associated with Moderna, even though that's one of the many, many, many things you accomplished. And yet, 3 years earlier, in May of 2020, there was a Boston Globe article that said, this is not how science should be done. Making a reference to your work when probably only 8 patients had been tried. So sort of the skepticism about your work has been kind of a hallmark of your career, and time after time after time, you overcame and prevailed. Tell us about that experience between 2020 May and 2023 May.
Well, thank you. Well, first, I mean, you're right. A lot of things that I've tried to do as a scientist, are probably been unconventional. And, and like even me being an engineer, when I started back in the 1970s, I went to work at a hospital and I was the only engineer there. And so I got ideas about how you could apply things like engineering to medicine, and that would ultimately lead to, a number of different treatments. In fact, at that time, one of the things I did back in the early 70s was develop the first nanoparticles that could deliver large molecules like RNA. And people were skeptical of that then. And actually, my first 9 research grants were turned down, and no faculty department in chemical engineering, which is my background, would hire me. So I give that as background to what happened to your question. So Moderna, I mean, it's of course a giant team effort. There just have been outstanding scientists there. But, I felt that Moderna was doing great work. We were in, 13 clinical trials, even when COVID had started. And so, to me, the science, even in May, as you accurately pointed out, even the dates, everything, I thought was very good. But there's one of the things that sadly I found in science and sometimes, investors, and that is what I'll call jealousy. A lot of people didn't want Moderna to work. I mean, even though it could have saved lives. And I think that that's a sad thing. And, the Boston Globe was only taking comments from different clinicians and stock analysts and so forth, when they made that headline. So, of course, what happened, I think between those 3 years that you mentioned is historic. I mean, the messenger RNA treatments have, according to various analyses, I think there's one of the major foundations estimated that it saved something like 18 million lives. It saved the economy trillions of dollars. And of course, now messenger RNA therapies are not only being used for COVID, but, there's, I think, very exciting, promising data for new cancer vaccines that are in clinical trials for melanoma, and many, many, many other treatments. So, and of course, from a financial standpoint, Moderna's stock, certainly compared to that early time, went way up. Now it's gone down some, but still, anybody that invested in Moderna obviously would have done spectacularly well. So I think that people now recognize that messenger RNA, therapies are important. And, for me personally, it's just been an honor to be a part of a company that has been so dedicated and has tried to help, and I think has helped so many people.
Right. Robert, I wanted to, Ajit took you back a little bit, and I think maybe we'll do a little bit of this. Ajit will keep taking you back to,
That's any way you want.
Yeah, this wonderful trajectory. I'm going to keep bringing you back to the marvels of the present. I was a little bit distracted with that liminal human stuff that was going on at the back. So I didn't share some of the really interesting statistics about Dr. Langer. I don't know if Ajit has already done that, but, he has 1,400 patents. He's, created 40 companies. He's the most cited engineer in history. He's literally, his epithet is Edison of Medicine. And also because he's married engineering into the life sciences. Bob, I would love you to speak about the fact that, you can do this level of precision delivery of medicines into the most difficult to get to cells. And you do that using wonderful materials. So can you talk to us about, in your own mind, what are the maybe 3 really breakthrough things that you've done where today you can legitimately say that your work has impacted 3 billion lives, So talk to us about the materials, which is uniquely you. What's the 3 most exciting things that you've achieved?
Wow, what an interesting and difficult question. Thank you. I think some of it goes back to, let me try to come up with, give you an answer to that on those 3. Well, the first one I'd say is actually what I mentioned back in the 1970s. That we developed, and this was published in the journal Nature in 1976, we developed the first material systems, actually very tiny systems, that could deliver large molecules. That went against conventional wisdom. And of course, that paper and of course the ones after it, often people
may
attribute
I just stop
to.
You for a moment? Can all of you hear Dr. Langer at the back? Can I just tell the console, there's a lot of reverb here, so we are not absolutely able to catch what he's saying. The same thing that happened earlier with the director's session. Sorry, Dr. Langer.
No. Should I speak louder? Would that be helpful?
No, the audience can hear you perfectly. That's totally
okay. I usually have a pretty loud voice, but let me see what I can do.
No,
so the 3 things. Well, I think the first would be what I mentioned, before, which was the ability to make tiny particles based on materials that could deliver large molecules. And that we published in Nature in the year 1976. We also use that approach to actually isolate the first what are called angiogenesis inhibitors, molecules that could stop blood vessels from growing in the body. And that would lead to many drugs, again, developed by others, but drugs like Avastin, Lucentis, Eylea, used by many, many millions of people around the world. So I would say that that's one. And obviously, the drug delivery work that would lead to many things, including the nanoparticles that Moderna and others would develop. Second thing is I would call more a paradigm shift. When I started doing work, in medicine, pretty much all the materials that people used in medicine were developed or derived by medical doctors. And what they did was they'd go to their house and they'd kind of find the organ or tissue, something that in their house resembled the organ or tissue they were trying to fix. An example I often use is for the artificial heart, they actually picked a lady's girdle material because it had a good flex life. But that would lead to problems. And like when blood hits the surface of that artificial heart, it could form a clot. The clot goes to the patient's brain, they get a stroke and they die. And I started thinking, rather than take materials from your house, you could really use chemistry to design materials from scratch that would have better and better properties. That ultimately would lead to new brain cancer treatments. And some of the other things we did lead to drug-eluting stents. So I would say that's probably the second area. And then the third area is probably the whole idea of creating tissues and organs from scratch. This was work we started with Jay Vacanti many, many years ago also, with the idea that you could take materials and combine them with cells, maybe even including stem cells, and that would lead to artificial skin. It's now in clinical trials for many different things like new blood vessels. And then it's also leading to what's called organs and tissues on a chip, so that you could really someday recreate different tissues. Like some of my students have made a heart on a chip, a gastrointestinal tract on a chip. The importance of that is, I think, severalfold. First, I hope it will someday reduce animal and human testing. And secondly, you could much more rapidly, theoretically, develop new drugs, new drug delivery systems. So I would probably say those three areas to your question.
I'm going to draw Ajit back into the conversation, but we had a bit of a scuffle in the beginning of the session, and we kind of made a little heroic biopic of you, literally in a minute. That's 4 decades of your life in 1 minute. But I do want to show that to the audience, Dr. Langer. So just bear with us for a minute. And
I'd like to see it myself.
Yeah, you're going to like one scene in it where you come on as a substitute for Tom Cruise, So let's show you that.
Definitely want to see that.
So
Wow,
even if
thank you.
virtually that's the treasure house we are speaking to, did you like that?
Terrific. I love a copy. Thank you so much. That's a tremendous job you did.
Right. Great. So, we're going to come back to some of those miracles we just showed on screen. I got the number wrong. It's not many people who can say their work has touched 4.7 billion lives. And, all the scientific community agrees that you have. So that's just amazing. And I wish that we'd been able to have you here in person. But Ajit, you had some other questions.
I'm going to pick up on the cue you gave me of taking us back in time. So, Bob, I'm going to take you back to the 27-year-old Bob Langer in 1976. So you finished your PhD in chemical engineering, and we were in the midst of this oil crisis globally. And if I understand correctly, you applied, or you were made to apply for many jobs in the oil industry, and you had some 20-odd offers that you declined because you didn't think that'll have the impact. Take us back to your time, and if you don't mind, walk us through your journey for a few years after that.
Sure.
What triggered your decision not to join those very lucrative offers that you got?
Sure. So I got my doctorate actually 50 years ago. I was showing my age. From MIT, and there was, as you pointed out, a gas shortage. And actually, if you lived in Boston, where I lived, you had to wait in line for your car to get gas, sometimes for 2 hours. The consequence of that is that if you were a chemical engineer, you got a lot of job offers. But most of it really, when I would go to these interviews, it was like increasing the yield of certain petrochemicals by very, very tiny percentages. And that, I guess, would have been worth a lot of money. And they did certainly pay very, very well. But I guess I just felt like increasing the yield of certain chemicals by like 0.01% or 0.1%. I just felt like I didn't want to do that with my life. I really wasn't sure what I did want to do, but I didn't want to do that. So, when I was a graduate student, I helped start a school for disadvantaged high school students in Cambridge when I was doing my thesis. And I developed, I think, new chemistry programs and new math programs. One day I saw an ad to do something like that at City College of New York and as an assistant professor. So I wrote them a letter applying for a job, but they didn't write me back. But I really liked that idea. So I found all the positions that something like that. I found about 40 and I wrote them and none of them wrote me back. So that wasn't going so well. So I kept thinking, what other ways could I use my chemical engineering background to help people? So I thought about medicine, which then for a chemical engineer would have been very unusual. I wrote to a lot of hospitals and medical schools. They didn't write me back either. So one day I was walking in the lab and one of the older scientists said to me, Bob, there's a surgeon in Boston named Judah Folkman. He said, sometimes he hires unusual people. He thought very highly of Dr. Folkman. I won't say what he thought about me. But anyhow, I wrote to him, and he was kind enough to hire me. And he had this idea, which relates to what I was saying before, that if you could stop blood vessels, maybe that would be a way to stop cancer. And I thought it was a visionary idea, but it turns out that he was widely criticized. So he hired me to prove he was right, and in so doing, isolate the very first blood vessel inhibitors. That's how I got started. I mean, it was very hard for me because I didn't have a biological background, but I guess I thought about things differently, and that's why I developed the delivery systems and ultimately was able to, we wrote papers in the journal Science and Nature showing that there were blood vessel inhibitors, and that also would lead to these tiny little particles that could deliver large molecules like RNA.
Dr. Langer, this actually plays back wonderfully to a discussion we've been having all day. You're literally the kind of, the ultimate example of this, that you were rejected by everybody because you were an engineer with no biosciences background. And so if there was an AI system that had been triaging your applications, you would have never got picked for a hospital job, But it took the individuality of one particular doctor to spot that and to actually enjoy a maverick, and so to be able to hire you. And that exactly speaks to what we've been talking about, the deficiencies of having AI systems hiring and firing and triaging applications, So in that sense, you're again a wonderful note to end the evening on. But the other question I wanted to ask you, that we just showed this, and the reason we wanted to put that film together is to bring home just how complex a human organism is, And in that, you've been able to create these precision drugs. I read somewhere that you've literally created something which is the molecular equivalent of a coffee cup, and that it releases drugs in phases, in ways that you can remote control outside. So again, as pure storytelling, will you tell us the marvel of what that is and how difficult it is to do what you and your team have achieved?
Yeah, well, that's a very interesting question. So, back in 2012, Bill Gates came to see me, and he, as you may know, is very interested in vaccines, and particularly vaccines for the developing world. And he said, well, could we ever come up with a way to create what we call self-boosting vaccines? So I have an outstanding woman scientist in the lab, Anna Jakulenik, and she and our team figured out a way to do that. I mean, it's still in animal trials, but basically we've created these tiny microparticles, and you could envision them as little coffee cups or little eggs with eggshells. And the idea is that you can put the vaccine inside each of those. But each of those coffee cups or eggshells, we can make break at a particular point in time, like 1 week, 1 month, 1 year, whatever time you want, by simply changing the composition of the eggshell or the coffee cup. And the beauty of it is what Anna and our team did is they changed it by taking a material, again, a polymer, that breaks down into water and carbon dioxide. So it's very harmless. And basically by changing the molecular weight of it and changing the ratio of different units, we can make it break at any time. So that's what we did. We came up with a new printing process to make these little coffee cups or eggshells. And then by changing the composition, you get these boosts at different times, but it's like a cocktail. You give the injection, and some break at time 0, some at a week, some at a month, and so forth. But it's a single injection. So that's really the idea. Anna and our team have tried it on 5 different vaccines so far in animals, and it works quite well. In fact, I know they're also talking to the Serum Institute in India about possibly certain collaborations. We've published this in the journal Science and several others as well. So that's a little bit about how it works.
That really does call for a round of applause.
So Bob, I want to build up on sort of the wide-ranging impact you've had on medicine. You made 2 references to the drugs Avastin and Tarceva. If my memory serves me right, Avastin was approved by the FDA in 2004-ish, I think.
Correct.
And then Tarceva for non-small cell lung carcinoma, maybe a year or so later. And both of these drugs had to do with this anti-angiogenesis, or stopping the growth of blood vessels. And it dates back to your work again in the 1970s when you were working with Dr. Faulkner. So that was a 2.5-decade journey from your early work on anti-angiogenesis and the FDA approval of these drugs. Would love to hear what it was like to working with Faulkner, and how your work ended up in cancer treatment with these macromolecules.
Sure. Well, Dr. Folkman was a visionary scientist. He passed away in 2008, but he was a visionary scientist, and he was a wonderful person for me to work with. And it was a struggle. I mean, because people didn't believe that this was a way, to treat cancer or really anything. But we did, like I say, publish this paper in the journal Science in 1976. We followed it up with several others, including ones in Proceedings in National Academy of Sciences a few years later showing this approach was safe. That was another objection that people had. But ultimately, we just got it started. There are companies, Genentech in particular, and the work of Napoleon Ferrara would be very, very important, identifying what's called VEGF and making antibodies to it, and really literally spending billions of dollars to get Avastin approved. And that, as you pointed out correctly, was 2004. That medicine is like that. Medicine normally takes a long time. I mean, the COVID crisis accelerated that for the COVID vaccine that Moderna and BioNTech developed, but most of the time, medicine is slow, and the people want to err on the side of safety, which is totally understandable. But that was really what happened, and we got it started, developed a number of the bioassays, and really proved for the first time that such molecules did exist and could exist.
So, Bob, I'd love to ask you, again, I'm kind of playing back into a lot of the strands that we've been discussing in the conference the entire day. We've been trying to decipher what is the essence of being human. And so that's why I'd like to ask you that, would you be able to kind of speak about what is the essence of being Dr. Langer? Is there a particular mindset, a psyche that has made you uniquely you and allowed you to do this kind of scale of work and run the kind of labs that you have students that created $40 billion companies? What is that unique, particular mindset of being Dr. Langer?
Well, that's a great question, too. It's probably better to ask my students, but what I think about is I've always wanted to see the things I do, whether it's teaching or science, do good. And, and that's one aspect. I suppose another aspect is I'm a risk taker. I'd rather try and fail than try to do something I felt would be really, really important and change the world and make it a better place. I'd rather try and fail than not try. I mean, and it sounds simple, but, so many people, when they graduate from college or whatever, they want a security. So that a lot of times people will do more incremental work, whether it's academia and just, publishing this paper or that paper. But, for me, I felt, and I still feel, failure's okay. And I certainly failed many times, but I'd rather try and do something that I feel will help many, many people, than just do something secure. And so I've taken chances, with my career, and I've certainly paid a price for it at different times. Like I say, when I tried to get my grants early on, my first 9 were rejected, and I couldn't even get a faculty position in the one area that I had studied, chemical engineering. But, in the long run, I guess it's paid off, and like you say, a lot of people have been helped by what we've done. And of course, now, that of course is what I wanted to see happen.
So just before I let Ajit ask you perhaps one of the last couple of questions, is that if you were taking on someone like yourself back when you were in your 20s, and one of your students had this, completely mad idea of doing the kind of materials implants into the human body that you were backing, would you have backed yourself at that age? And what about your proposition would have caught your attention?
Well, I hope I would have. I like to think I would have if I, had experience. I've certainly, in my career, I've taken very unusual people, who don't exactly have the ideal background, if one was looking at what our lab does. But those people, 2 examples are Gio Traverso, who's a molecular biologist and gastroenterologist, and Dan Anderson, who's a molecular biologist, and both of them are now professors at MIT, but they came to my labs as fellows or postdocs, but they had very different backgrounds. I think it's because they had very different backgrounds that they've become such great leaders and have done such wonderful work. And I've gotten plenty of other people as well like that. So I hope that people had seen, that I, had a different background, and I did have had won some honors for different kinds of things, and I hope that they would have, I would have taken a chance.
Right. Ajit, sorry, because it just literally segues into this, that I read somewhere that your lab is full of atypical human minds, Is that a good description of your lab? And does that only mean atypical in terms of backgrounds or atypical in the way of thinking? And is that an accurate description of your lab? Well,
I might phrase it differently. I mean, some people could say that. I would say the best description to me is it's very, very interdisciplinary. We have people probably with 10 to 15 different disciplines, all of whom I think are extremely bright, all of whom I think are driven by this mission of doing curiosity-driven science, but not just leaving it there, but want to see it, be done to make the world a better place. So I think about it like that. That may be atypical, but I think that describes a lot of the people that are in our lab.
So, Shoma, actually, I'm going to build on what you were asking earlier, the essence of being human or essence of being Bob Langer. And just in the previous session, you were discussing with the speaker, should we upload our totality at the end of our life, somewhere in the beginning of our life, and what are those two things? So I'm going to turn that question back to Bob. And so two related questions, what it means to be Bob Langer in, say, 1979, or let's pick 1983 when your first patent was published. And my second question will have to do with your present day yourself, but more on the personal side, not so much on the Bob the scientist, but Bob the human being. Your first patent was published in 83, and that was quite a journey to get there. There was skepticism on the part of the examiner, and some esoteric reference by some chemists to your work a few years earlier ended up playing the wild card to get that patent finally approved. Tell us about that story.
Yeah, well, that's a very good question. So when we did file our first patent, it got rejected 5 years in a row. And actually the people the lawyers and everybody told me I was wasting an enormous amount of money and it would never get allowed. But when we started this work, I mean, many years before, everybody told me it was impossible, it could never work. So what I did, I started to think, well, how could we convince the examiner? So I did a literature search, and you can do what's, like I mentioned, we published this paper in Nature in 1976. So now the 5 years of rejection were in 1982. So I did this literature search. I found a paper that said just that the work that we'd done was extremely surprising. And we showed that to the examiner, and he said, gee, he didn't realize that. And he would allow the patent if I could get written affidavits from the 5 very famous scientists who wrote it, that they really wrote it. And so I wrote them, and they were nice enough to write me back. They really wrote it, and we did. We got a very broad patent.
So the word surprising turned out to be in your favor in that case. It could have also gone the other way.
Yeah, it certainly could. And obviously, it did go the other way earlier, because that's why I got all my grants rejected and couldn't get a faculty job. But you're absolutely right. You can. In fact, Dr. Folkman once said to me, he said, I want to save all my grant rejections, because using your strategy, I'll be able to send them to all these patent examiners.
Right. Dr. Langer, we are kind of out of time. We've had a very long but exciting day. I just want to leave the audience with, we've already talked about your marvelous journey, and I'm glad that this is just such a tiny introduction to your work, because I hope next year there's going to be no scheduling issue, no sudden meetings, and we're still going to have you here in person. But I'd just like to leave the audience with the thing that you just mentioned in passing, that you're now creating patients on a chip, and pushing the frontiers of medicine to some next level. Earlier, it was just organs on a chip. You were growing skin on a scaffold. Now you're growing many organs on scaffolds and looking at the impacts of medicine on one organ and its cascading impacts on this adaptoid that we call the human body. You're the master excavator of that body, the mapper, the cartographer of that body. And you've, like I said earlier, improved 4.7 billion lives, made it easier. So for all of that, a big salute to you, and thank you very much for joining us. Thank you.
May I take one more minute, if you don't mind? So Bob, the audience is already giving you an applause, but I'm going to interrupt that applause. You are a human being. You are a person. You have a family. You have a wife. You have 3 children. 3 adult kids. Would love to hear that part of the story. What does it mean to be your family?
Well, I'm very lucky. My wife got a PhD at MIT, and so now she's doing more art, but she's very smart, and she's probably a lot smarter than me, and so she's very understanding about my life, and the children are all doing well. They're now all in their 30s. In fact, I've got one grandchild now and another on the way, and one is developing, has worked in biotech and is starting a venture capital fund. That's my oldest son. My daughter is actually, she's the president of a biotech company and that I'm involved with. Also, she's on the board of Biogen. And like I say, we have one baby daughter and another baby daughter coming. And my youngest son, Sam, is married, living in California, actually trying to do a lot of good for people. He's involved in innovative farming and other kinds of things. And so but I'm very proud of all of them. And, but I owe my wife a lot, she's a very straight, I always know where I stand. And, and she's been very understanding of my life, and, has just been a great person.
Beautiful. That's a wonderful note to close on. Thank you.
I did want to share just one more last anecdote, and this is really the patience of the audience, that again, Michael Sandel was trying to arrive at these qualities that make one human. So I just wanted to share with you that when I was trying to reach out to Dr. Langer, I kept waiting for introductions to him, even through Ajit and through others. And then one day I finally just wrote to him, and it took him all of, I was thinking, okay, he's a very famous man, he's a legend, he's a legendary scientist. I need an introduction to get to him. And then I just wrote to him, blind, and I heard back from him in 10 minutes, literally by the clock, 10 minutes, saying, this sounds wonderful and I'd like to come. And when the schedule came up, he actually said, the busy man that he is, he said, book me a flight on a Friday, you can get me there on a Saturday and get me back to Sunday. I'm willing to make the journey, It's not very often that we get very busy, very successful people to have that humility and compassion. So again, a big salute to you, Doctor. Thank you very much. Thank you.
Thank you.